Hi biologists, let's start by looking at the learning objectives for this lesson. At the end of this section and following the biology syllabus, you should be able to understand the term ultrastructure and be able to identify and know the function of the cell membrane. What does this actually mean? What are we trying to understand? Well, it's straightforward enough. You have to understand the term ultrastructure and you have to be able to identify and know the function of the cell membrane. Let's sort cell ultrastructure by looking at a few ideas that we met before. An organelle is a structure in a cell. A nucleus is an example of an organelle. Most organelles are colourless or transparent, and they must be stained in order to be seen under the microscope. Plant cells are usually stained with iodine, whereas animal cells can be stained with methylene blue. Now, what do we mean by cell ultrastructure? Well, ultrastructure is the detail of a structure as can be seen using an electron microscope. Now don't forget, electron microscopes are a lot more powerful than light microscopes. So obviously structures are seen using the electron microscope that were hidden when using the light microscope. Let's look at an animal cell under the electron microscope. Notice the shape of this animal cell is different to the shape that we drew in the previous video. Cell shape is totally irrelevant. Cells have different shapes and sizes. What we need to focus in on here is we need to realize that the cell components that we saw already are going to be present here and can be seen under the electron microscope. We labelled the cell membrane already. The cell membrane is the skin of the cell. It is semi-permeable. It acts as a sieve. It controls the passage of substances in and out of the cell. It will let small molecules or chemicals like water pass in and out freely, whereas it will prevent the passage of larger chemicals. We also met the idea of cytoplasm, the watery jelly, the site of chemical reactions, the nucleus, which controls the activities of the cell, and sometimes animal cells have small vacuoles. Now, as a trick, I like to label the diagram like this. Cell membrane top right, cytoplasm top left, small vacuole next, nucleus on the left. Now let's include the cell components that you would see under the electron microscope and that we did not see under the light microscope. The first cell component that can be seen is the mitochondrion. Now cells have loads of mitochondria, plural. However, for the sake of the exam, we will just put in one. The next cell component is a centriole. A lot of biology books don't include the centriole in the animal cell. I like to include it. It is present in animal cells and we will need it for the understanding of a future section. The next cell component are the ribosomes. The ribosomes can be scattered throughout the cytoplasm or attached to the last component that we're going to mention here. The last component is the endoplasmic reticulum, and the ribosomes can be attached to the endoplasmic reticulum. Quite often, the endoplasmic reticulum is continuous with the nucleus and found in cells that secrete fluids. What about the plant cell under the electron microscope? Again, we should see the parts that we already saw using the light microscope. And here we will make use of our drawing trick. What was the first label up on the top right? You got it. The cell membrane. Exactly the same procedure here to make life easy. Remember, the cell membrane is semi-permeable. What was the label up on the left? The cytoplasm, the site of chemical reactions. Then the nucleus. What was the label on the right? It was the vacuoles. Here, however, because we are looking at the plant cell, then the vacuole will be a large vacuole. Now let's start putting in the other organelles. What was in the middle on the right? The mitochondria or a mitochondrion. And over on the left, this time, instead of a centriole, we will put in a chloroplast. Chloroplasts are only found in plant cells. Remember, they are the structure that contain chlorophyll. Just to point out, plant cells have loads of chloroplasts. Again, we are only going to put in one for the sake of the exam. What was in the lower right? Can you remember? Oh yes, the ribosomes. The ribosomes make protein for the cell. They can be found free in the cytoplasm or attached to the last component, the endoplasmic reticulum. Don't forget, because this is a plant cell, we have an extra label. Extra component is the cell wall, made of cellulose. Cell walls are present in plant cells, but not in animal cells. One of the objectives of this lesson is to be able to identify the cell membrane. So let's have a look at the structure of a cell membrane. The structure of a cell membrane reminds me of a two-ply tissue. There are two layers in a tissue and you can pull them apart. The cell membrane is rather like that, except the tissue is made of what we call phospholipids. There's a top layer and a bottom layer of phospholipids. Hence, we call it the phospholipid bilayer. Embedded in the phospholipid bilayer, you have protein. The proteins can be removed or inserted without making any difference to the overall structure of the cell membrane. This is why the structure is rather fluid. Because the cell membrane is semi-permeable and acts as a sieve, then of course the cell membrane will have to have pores in its structure. So in a nutshell, what is the structure of the cell membrane? Well, all biological membranes. It doesn't matter whether they are cell membranes or membranes around chloroplasts or membranes around nuclei. All biological membranes have the same structure. The structure is referred to or called the fluid mosaic model. In this structure, the cell membrane has a double layer of phospholipids, a phospholipid bilayer. Proteins are embedded in the phospholipid bilayer. Detachable proteins can move throughout the bilayer without affecting its overall structure. What's the function of the cell membrane, also called the plasma membrane? Well, as we've said before, the cell membrane controls what enters and leaves the cell. This is absolutely vitally important. You must understand that cell membranes are semi-permeable. The cell membrane helps to hold the cell contents. It acts like a plastic bag holding watery jelly. Sometimes the cell membrane can recognize foreign particles or sense molecules that touch them. But we won't lose any sleep over this function. Let's look at a picture of a cell membrane as taken under the electron microscope. Such a photograph is called an electron micrograph. Be able to identify the cell membrane in such a photograph. There you have it. Now the best approach to this is to make sure that you are able to draw large, clear, well-labelled diagrams of all of these structures and know the functions of each part. Practice in a jotter. Now that we have reached the end of our lesson, have we achieved our objective? Can you understand the term ultrastructure? Can you identify and do you know the function of the cell membrane?